Welcome to the course on basic genetics. There are three teachers in this, Dr. Richa Are, Professor Madhu Gavaldas Tapadia, and myself Jagat Kumar Roy. We are from Department of Zoology, Banaras Hindu University, though I am retired. This is week 11, lecture number 1, and the title is Human Genetic Disorders, Chromosomal Aneuploidy. We have seen human chromosomes in earlier lectures. Human chromosomes, diploid number is 46, and this is a karyotype. You have seen the karyotype. Once again, you can see the karyotype that we have divided all chromosomes into 7 groups, A, B, C, D, E, F, G, according to the size and according to the shape of the chromosome. The right side, you can see the G-banded chromosomes and the karyotype of G-banded karyotype. So, in these chromosomes, the 46 chromosome number, the karyotype is now, I am sure, clear to all of you. We have already understood that cells of normal individual can have either one complete set of chromosomes, that is haploid. For example, 23 chromosomes in human coming from one gamete, or 2 complete set of chromosomes, that is diploid. Example, 46 chromosome number in human, the picture that we just saw. And if a set of chromosome is incomplete, with one or a few chromosomes missing or duplicated, then this is termed as aneuploidy. Let us discuss today human chromosome aneuploidy. Once again, this picture is known to you. This is Down syndrome, and here you have the trisomy for chromosome 21, which is here. So there are 3 copies of chromosome 21, and this results in certain features as you have already once saw, and the main features are they have a mongoloid face, that is round face, the epicanthal fold, that is fold on the eyes, and the single palmar crease, that is one crease, not like the crease we have, the one crease that is there. We have also discussed extra X chromosome, that is 47 XXY, a male, or 48 XXXY. These syndromes are called as Klinefelter syndrome, and there again there are some of the anomalies that you have seen: tall stature, tendency to lose chest hair, poor beard growth, breast development, female pattern of distribution of the pubic hair, and long arms with legs, testicular atrophy, and so on. Beside these, let us consider some other chromosomal anomalies. Chromosome 18 trisomy. We have seen chromosome 21 trisomy. We have seen chromosome trisomy in chromosome X, that is XXY so far we have seen. Now in Edwards syndrome, chromosome 18 trisomy is there. In this, there are certain features. They have congenital heart defects, growth retardation, dysmorphic features. You can see dysmorphic features here. You can see the legs, see the limbs. They are dysmorphic. There is a facial cleft, and very importantly, spina bifida, that is this thing. So what happens that the spinal cord, inside you have the neural tube, and then the spinal cord is all around. The neural tube is closed. Initially, this is an open tube which is closed from both the ends. But here, those closures are not taken place. In spina bifida, some of the spines are defective. Result is the spinal cord that comes out of the body and it projects like this. Severe developmental delay. So these are because of chromosome 18 trisomy. That means in place of 2 chromosomes, if there are extra chromosomes, that is 1 chromosome in 3 copies, that causes havoc. And the incidence is 1 in 2,500 pregnancies. They die. Another syndrome, that is Patau syndrome. Here, chromosome 13 trisomy is there. That is chromosome number 3, you can see in 3 copies. What are the features? Severe intellectual disability, developmental anomaly, and most die within 1 year of life. This is 1 in 10,000 or 20,000 live births. Some of the features, one of the very important feature is lip or cleft palate, low set ears, and small head. So these are some of the features. That means chromosome 18. Initially, we have seen chromosome 21. We have seen extra X chromosome. We have seen chromosome 13 in 3 copies. Also chromosome 18. These things we have seen. Now let us come to another syndrome, that is Jacob syndrome. Here, chromosome number is 47 and there is XYY. That means 1 extra copy of Y. Earlier I had discussed that Y chromosome is very small, X chromosome is considerably large. Y chromosome contains fewer genes and it contains essentially those genes which are responsible for human fertility. So here the individuals are tall, muscle weaker, learning disability, behavioral anomaly. Incidence is 1 in 1,000 male birth. This is a male because this has Y chromosome. And most lead normal life and they are fertile. You can see a great difference. So far we were seeing chromosome 21, chromosome 18, chromosome 13, where 1 chromosome extra, that causes lot of problem. Now, 1 Y chromosome is extra, the problem is minimal because there are only fewer genes. That means imbalance of only a few genes. Let us come to triple X syndrome. This is another syndrome where the chromosome number is 47 and this has XXX, that means 3X chromosome. You can see here 3X chromosomes. This is female and the individuals are tall, learning disability, often remains unnoticed. The 3X chromosome many a times remain unnoticed except the females are a bit tall and incidence is 1 in 1,000 female births. So, regarding sex determination, we did talk once that if there is Y chromosome, if there is presence of Y chromosome, the individual is Y. If there is presence of X chromosome, the individual, presence of 2X chromosome, no Y, individual is female. So, here in human, Y chromosome essentially is sex determining. But what happens when there are more than 1 X? Let me talk it like this, that in male, we have 1 X chromosome. In female, we have 2 X chromosome. For the time being, just do not consider Y chromosome because it has just few genes. So, in female, 2X chromosome versus in male, 1X chromosome, there is huge disparity. That means females, for X chromosomal genes, X chromosome contains a large number of genes. So, females contain all those genes in 2 copies, male contain in only 1 copy. That means there is a dosage problem. Both of them, because it is not that X chromosome genes are only for femaleness or only for fertility, nothing like that. So, nature tried to balance the 2. This is called as dosage compensation. By dosage compensation, what happens? In female, one of the X chromosome, out of the 2 X chromosome, 1 X chromosome randomly gets inactivated. When it gets inactivated, this becomes heterochromatinized or appears like a dot, which is known as bar body. Bar is not B-A-R, but B-A-R-R, because this is the name of a scientist bar. That means human body and this X chromosome inactivation is random. One of the X chromosome has come in female from father, one of the X chromosome has come from mother. So if the dosage has to be compensated, one of the 2 X chromosome randomly get inactivated. But this inactivation is not at the 1 cell stage, much later than that. That means if there are several cells and if X chromosomes are randomly inactivated in female, that means in one, it is possible that mother's X chromosome is inactivated. In another cell, let it be that father's X chromosome is inactivated. So, in such conditions, female body is a mosaic body. That means having patches of male X chromosome inactivation, female X chromosome inactivated. So now, with this information, when we have 3 X chromosomes, so far we have talked that we have only 1 X chromosome, 2 Y chromosome. When we have 3 X chromosome, then this will have 2 X chromosome inactivated and nature will try to balance that. So these individuals often remain unnoticed. This we had seen earlier, that this is Turner syndrome, where the individual contains only 1 X chromosome. Another X chromosome is missing. You call it as XO. O means 0, no X chromosome. So here we have already discussed some of the features. Low set ears, this is not sit, this is low set ears. Small jaw bones, short and wide neck, cardiac problem, swollen hands and feet. The gonads are rudimentary, infertility, white chest with nipples far apart. So these are some of the problems. So just now we were talking about whole chromosome, either extra copy or 1 copy less. Now let us come to some other. This also we can consider as the chromosomal anomaly, but this is deletion of part of this chromosome. Chromosome number 1 is long. This is the longest chromosome. And here 1p36 region is deleted in this syndrome. The syndrome name is 1p36 deletion. Just to remind you once again, the 2 arms of a chromosome, you call it as P arm and Q arm. So this side is P, this is the centromere. From centromere, this is P and this is Q. The numbers also go in the same way. So I was showing you this chromosome. Chromosome 1, the longest chromosome, the longest metacentric chromosome. Here, where is P36. It is not 36, never pronounced as 36. This is P36. That is in P arm, band 1, 2, 3, and in that sub-band 6. So this is the region. The deletion of this causes congenital genetic disorder with moderate to severe intellectual disability, delayed growth, seizures, limited speech ability, malformations, hearing and vision loss, distinct facial features, and this affects one individual out of 5,000 to 10,000 births. Let us come to another one. This one you have seen. This is cry of cat or Cri-du-chat syndrome. This is due to deletion in 5p region. So again, this is chromosome 5 and p region is this region. And here chromosome 5p, if deleted, the symptoms, this occurs 1 in 20,000 to 50,000 live births. The symptoms are high-pitched, cat-like cry or weak cry due to muscle weakness. The cry of human, when the muscles are strong, the cry is loud. But if somebody cries like cat, that is meowing, that means muscles weak, they cannot cry loud. Low birth weight, small head, rounded face, broad flattened bridge on the nose, that is the nose is flat, eyes spaced wide apart, folds of skin over eyelids. This fold over the eyelid, it is on both sides, and this results eyes looking more circular round than the normal shape of the eye. Folds of skin over the eyelid, this I just talked, and then abnormalities of palate such as unusually narrow and high palate. The palate is quite high and this is narrow. So these are some of the symptoms of Cri-du-chat or cry of cat syndrome. Then we come to retinoblastoma. Again, quickly, we did talk about retinoblastoma. This is due to deletion of Q14 region of chromosome 13. So this is chromosome 13. Here Q14 is, this is Q1, then Q14 is here. This is Q14. So this region, if it is deleted, this causes retinoblastoma. Now see here that if such kind of deletion is there, and if the deletion is in heterozygous condition, that means one chromosome has normal 13q14 region, the other chromosome has deletion. What will happen to such heterozygotes? Nothing will happen, because one chromosome, this region is sufficient, this region, the gene name is RB, this is sufficient to make the required amount of RB protein, and so the individual is happy. If the deletion is in homozygous condition, that is on both the chromosomes, then this causes the problem. This causes the retinoblastoma, that is the cancer on eye. After going through this, I'll once again ask the question, why the cancer is in one eye, why not in both the eyes? So, what is the mechanism? What this RB gene does? This RB gene, during cell cycle, during G1, this holds one of the transcription factor and does not allow to function. This results remaining in G1 and cell cycle cannot progress further. So this is called a tumor suppressor gene. That means cell division cannot occur if this is present. Cell has device. When it has to divide, this has to be phosphorylated so that this is released, and then the function of E2F is done, and again this will be dephosphorylated, again this will catch. That means for time being, there will be release of E2F that nature has devised. But if suppose this is not there, if suppose homozygous deletion of this is there, so this protein is not formed at all, result is cell cycle cannot stop. Cell cycle will continue, and that leads to cancer. Once again, I'll go back to this picture. There are two questions I'm asking. If this deletion in homozygous condition, that means the whole body is having the deletion in homozygous condition. That means this important tumor suppressor is not being formed. So both the eyes should be having the cancer. Both eyes are equal. Why in one? This is a case where the individual is heterozygous, not homozygous. If it was homozygous, both the eyes would have got this. Because this is heterozygous, in one eye, in one or more than one cell, another mutation that deleted or that inactivated the retinoblastoma gene, the lone copy that was there because of heterozygosity, now that is deleted or that is inactivated. This resulted in retinoblastoma of one eye. So this is the explanation for unilateral versus bilateral. Another thing I'll ask, that if this is one of the important tumor suppressor gene, then throughout the body there should be cancer. Why in eye only? Always genes may have some preference for its tissue specific expression. More than that, it is not the only tumor suppressor. There will be a coordination of many other genes along with this. So eyes, because there at early age the cells are dividing, that gets cancerous. If these eyes are removed, then the child develops cancer later in other parts of the body also. So I'll go to one or two more, that is Prader-Willi syndrome. Here there is a small part of chromosome number 15 is deleted. So this is a rare genetic disorder, 1 in 10,000 to 30,000 individuals due to loss of function of specific genes on chromosome 15. A part of chromosome 15 is deleted. Symptoms in newborn: weak muscle, poor feeding, and slow development. In childhood, affected individuals become constantly hungry, leading to the obesity. The picture you can see, and this results in diabetes also, type 2. Affected individuals have narrow forehead, small hands and feet, short height, and light skin and hair. Also intellectual and behavioral anomaly, and most of them are infertile. So with this, I come to the activity questions and discussion. The question is, do we find trisomy or monosomy of all the chromosomes? Or there are only discrete example of chromosome 21, chromosome 18, chromosome 13, and part of chromosome 1, part of chromosome 13, and so on. Why not all the chromosomes having trisomy? There are genes on chromosomes, and these genes, if suppose the number is not adequate, that means if there is a disbalance in number of genes, that results in problem. The larger the chromosome, more genes are there, so very little chance of having trisomy or monosomy in those chromosomes. Smaller chromosome, there are more chances, And that is why chromosome 21 trisomy survives. Chromosome 18 also survives to some extent. Chromosome 13, as we are going towards the larger chromosome, they do not survive. The other question is, how can they get cured or how can they be cured? The answer to this is because these are chromosomal disorders. These are syndromes. These are complicated, a large number of diseases together. So they cannot be cured. The only thing is they can be managed with certain specialized trainings and other things, if possible. The other thing is the scanning before birth and termination of pregnancy if such kind of cases are there. Thank you very much.